So what I want to do is talk a little bit about some of the role of surgeons in clinical trials. I'm going to touch a little bit about phase 0 studies, but I'm going to mostly talk a bit about surgical trials and tissue and obtaining tissue for other studies. And here are my declaration of my conflicts of interest. In 2012, the National Cancer Research Institute in the UK published a document about the role of surgeons in oncology research. And they produced this document of the roles a surgeon can be involved with research. And they are, some of it is very much requires a surgeon and more surgical research. Others can do work across other disciplines. Some of it is also contributing to other research, some is leading research. And I'm going to really focus above the line here, the top part of this, where you require a surgeon. And what I want to talk a bit about is collecting tissue samples for research, and then about surgical research and innovation and how to evaluate new technologies. So this is a common thing we get as surgeons. Oh, we just want some glioblastoma tissue for research. Can you just give us some? Or can you give us some samples from your tumour bank that you've already got? And this is really difficult because one of the things about tumour sampling is you need to know where it's come from. It's all well and good giving you a bit from the centre of the tumour, but if it's all necrotic tissue that really has got very little viable tumour in it, it's not going to really help your research. So we have a series of techniques we use that can help us take samples from a tumour that can give us a bit more information about where that sample has come from that will aid your work and research. So the simplest, of course, is using image guidance. We do image guidance and image guided biopsies a fair bit. And you can even, there are some studies where you will do this before opening up into a craniotomy and resecting a tumour. And this is an example from a study we did some years ago, just doing the screenshot, that we can get very accurate biopsies using this technique. And it is very good for image tissue correlations. The problem with it is you get tiny specimens, and the specimens are often from these biopsy needles are so small that it may preclude doing a lot of work with them. It may be you can only really look at histology of these samples. And this is an example of our study. So this is a, let's jumped, oops, it's jumped, there we are. This is the example of our study. Where we were looking at trying to correlate FLT PET, fluorothymidine PET, with as a marker of a proliferation in tumours. And you can see here are the PET images with our biopsy sites at different sites. And this is the MIB1 stain looking for cellular proliferation. And using this very accurately, we could then quantify the uptake of the FLT and also look at the proliferation indexes of the tissue and show that there was a correlation. What are other techniques if you want to get larger samples? Well, this was a technique that was developed by my colleague Colin Watts in Cambridge and it was published by Sarah Piccirillo a few years ago now. And what they were looking at different areas using 5-LA fluorescence guidance within these tumours. Now, 5-LA fluorescence is a common technique used for resecting of glioblastomas. And the idea is that active tumour will take up the 5-LA and convert it to a protoporphyrin which fluoresces under a blue light. And what they realized was you could get different components of a tumor. So there's the area you can see here that looks pink, that is the fluorescence tissue. And when you look at that under the microscope, that is the features you see of an aggressive tumor. So a solid tumor with microvascular proliferation and very small areas of necrosis within it. Within the tumour, you can also find areas that have no fluorescence, and these tend to be the more necrotic areas where you get much more inflammatory cells and more necrotic material. And finally, once you resect the tumour and you get to the edges of the tumour, you also can find areas that don't fluoresce, and these are from parts of the tumour margin, and these will have variable amounts of tumour cells within them. And you can use this for, as you've already heard, for the window of opportunity study. So if you have wanted to look at you dose a patient with a drug and you want to see whether the drug is the delivery of drugs to the various regions, you can take samples from the fluorescent tumor core. You can also take some samples from the tumor margin. And if you needed to, there's also no reason you couldn't take it from what appears to be normal brain outside of the area where you're operating. There's always something for certain neurosurgeons can always provide is the degree of normal brain, because often we have to go through normal brain to get to tumors when we are operating. The question really is which of those do you want to use as your target? And as you heard earlier on, that you may want to take multiple samples to look at uptake of drugs in different regions. As a very simple surgeon, I have my view on this is that the fluorescent core may not be the best part of the tumor to look at, because between these two images, we've taken away that area that was fluorescing that we've sent, that we may have taken samples from, and that sample is now sitting in a pot in the pathology lab. And it may be the tumor margin might be a more interesting part, because it will have the residual tumor that we should be looking at. One area that my group's been very interested in is using imaging to better identify regions within tumors. So we know that using contrast enhanced T1 contrast imaging and using something like FLAIR are not accurate. They're not accurate for identifying the full extent of the tumor. They also don't tell us very little about the pathology because they are effectively non-specific markers. So we have gone to use markers that tell us a bit more about pathology. And in this case, this is work done by Chao Li and my group, who used perfusion imaging, so took some RCBV images and also some ADC images, and tried to identify regions within the tumor where there was low RCBV, and then found corresponding areas where there was high ADC and or low ADC, and then combine the two to have these two regions of interest that we could then study and understand what was happening in these different regions. And we came up with this concept where we could identify some regions which had low cellularity, so in other words, they had a high ADC and low perfusion, and these all fitted with necrotic areas. And we also had other areas that had high cellularity but low perfusion. And this would fit with what we understand or what we believe is the stem cell niche. And it's work we're doing at the moment trying to confirm that that is what we're identifying in these regions. And similarly, we're looking at the other way around. Another student of mine, Natalie Bunzea, looked at the combination of high cellularity and high perfusion. And you can see these are the areas you can see here that are in pink. And you see those both in the contrast enhancing part of the tumor as well as the non-enhancing part of the tumor. So they are not restricted to one or the other. And if you look at the behavior of those, and we used MR spectroscopy as a marker of tumor in those regions, you can see that you have very similar results in terms of the choline to NAA ratio, which is a marker of cellular proliferation in both of those regions compared to the other areas around it. And what we're now able to do is to import this into our image guidance system, so actually we can see this during the operation. We can see these areas of different regions, and I'm putting this all in inverted commas because we're still trying to ongoing studies to confirm that this is what we're really seeing. And it may be that this may be a better way of identifying tumor samples or for sampling tumors in the future. Okay, so let's move on now to the other part I wanted to talk about, which is the bulk of my talk, which is about surgical research as itself. And this editorial in the Lancet by the Lancet editor Richard Horton really caused a lot of concern and a lot of browbeating amongst the surgical academic surgical community. And what he did in his editorial is it was related to a randomized controlled trial of a surgical intervention. But he actually said this is really good because if you look at he went through a number of journals, of surgical journals, and showed that the majority of the research that was published in them was case series, just a series of cases. And he said that this is the wrong way forward and the things should be properly evaluated. And this is a quote from a medical statistician going back to the 1920s, who said they would like to shame surgeons out of the comic opera performances which they suppose are statistics of operations. And they said that only when the quality of publications in the surgical literature is improved will surgeons be able to rebut the charge that as much as half of the research they undertake is misconceived. So this really caused a lot of concern. And if you think about it, surgery is complex. It's a complex intervention, and its outcomes are dependent not only on the intervention itself, but also on the operator, the team, and the setting. And there are some specific difficulties associated with these complex interventions. Learning curves are a big problem. The more you do, the better you get. And if you're introducing something new, it will take a while before people are ready to be able to use this. Quality variations exist, and also there's problems with the perception of equipoise. Now, throughout the rest of my talk, I'm going to use the term surgery, but as a complex intervention. But I should highlight that you could change that word and put radiotherapy or rehabilitation. And I'm going to show examples of both throughout my talk as well. So we've heard about the drug phases, the drug trial phases, and the stages of clinical trials. And with the issues we've discussed about the problems with surgical trials, this really doesn't work as a method of being able to directly put them across onto these phases. We need to think of another way of doing this. So this is where the ideal framework came from. It was a series of consensus conferences that were run at Balliol College, Oxford, between 2007 and 2009. It's often called the Balliol report that first came out of this, where they discussed some of the issues and came up with a framework to discuss the stages better and propose some recommendations of how this should be used. One of the problems is, what do you mean by a surgical innovation? So if you're trying to improve, if you look at the title of the final report that was published a couple of years ago now, no surgical innovation without evaluation. Well, what is a surgical innovation? And the problem is there is no consensus of what it means. And there's a variety of things it could refer to. So for example, it could refer to what effectively is a brand new procedure. So for example, the introduction of laser interstitial thermal therapy. Or what about if you adapt an established procedure, for example, resecting tumours, but using an endoscope to look around corners? What about small changes, which all surgeons do all the time, so-called tinkering, where you change what you do a little bit by making small changes, or the use of new devices, for example, using a new, for example, a dural substitute that you may want to use and see whether that has any difference for your patients. How do you? What are these that really are innovation? One of the other problems with adopting surgical innovation is the fact that there is the problem of often the problem of equipoise. So, and this is a law that was sort of set up as a bit of a comical thing, but actually it's got some truth in it. That it's always too early to do a randomized trial in surgery until, unfortunately, it's suddenly too late. And by that I mean, in the early days, there are so few people who are involved with these. Have the experience of using these techniques that it's too early to do a trial, and then by the time large numbers of people have taken on this innovation, then everyone is convinced that this is the best way forwards and it shouldn't go back and doesn't need exploring. So this is where the ideal framework from the collaboration came from, and it involves 5 stages: an idea stage, stage 1, a development stage 2A, an exploration stage 2B, an assessment stage 3, and a long-term follow-up stage, stage 4. And you can see that ideal is used because the first letter of these words. So let's just go through the stages one by one and sort of explain what you're expected to do at each of these stages. So this is the idea stage, stage 1. And this is basically the first in humans study. It needs to be registered so that there's a database that we have information about these patients, and it tends to be, it is a single patient operated on by a single surgeon in a single hospital. It's a case report effectively. It's designed to be a proof of concept, and it is advised that it is reported as a structured case report. Going on from that then is the development stage, or stage 2A. And this again may involve a smaller number of patients, may involve 1 or 2 surgeons, but tends to still be based in 1 unit. And the idea of this is to work out what is the optimum technique. What is it that you're going to do that you're actually going to then be able to evaluate at a later time scale, and also get an idea of which patients you should be using this intervention in. And the data you collect from this is predominantly safety data and some efficacy data that will help you power further studies. And the way this is done is through a prospective development study. And in fact, the most important part of this outcomes for these studies is actually the development of the technique rather than any of the other outcomes I've mentioned. So what is a prospective development study? Well, this is a study that has a prior protocol. It has a clearly defined outcome measures, and it's transparent reporting of cases. So basically what you do is you report, if you say, for example, you do 15 cases as part of this prospective development study, you report 15 cases one after the other, and you talk about how things change and where things change during that procedure and what effect that making those changes had. And it is these changes in your indications and your technique which are really the important thing for this kind of study. And one of the things that the ideal collaboration has recommended is that these techniques and devices in the development phases, and they're working with journals to positively discriminate in favor of these prospective studies, and to stop the use of accepting studies on a retrospective data, so the usual case studies that we tend to publish, except when there's no viable alternative exists. So by the end of that stage, you should have a method, and a method that can be taken to other units and other surgeons. And this stage, the exploration stage, is where you start using it in more widespread practice. It's again, larger number of patients, more surgeons are involved, and this time it is a multi-center study. And the questions you're looking at here is there a consensus on equipoise on a trial question, and also looking at learning curves and trying to understand learning curves. And the way this can be done is either through a prospective collaborative observational study or a feasibility randomized controlled trial. And here's an example of work that we're doing at the moment, looking at a rehabilitation technique, it's a home-delivered rehabilitation technique that we have taken from, has been developed for patients with brain injury that we are adapting for a neuro-oncology patients. And we have set it into 2 stages. The first stage is the ideal 2A study, where all patients will have the intervention. And we're basically learning how we deliver the intervention and what's the best way of doing it. And at the end of this stage, we hope to have a technique, and also we'll have some information about whether or not we think we can use this intervention in patients. And we'll have a go, no-go decision made at the end of this stage. Stage 2 then is this stage 2B study, where we have a feasibility randomized controlled trial, and patients then will be recruited and then randomized to either this so-called FACES intervention, which is this rehabilitation tool that we're using, or a placebo intervention, which is a general brain training app that they can use. And interspersed through our study, we have qualitative studies. And the idea of these qualitative studies is it helps you to work out how you're going to run the next stage of the study and how you're going to develop things in the future. So this jump between this stage 2B, which is this exploration stage, and the assessment stage, stage 3, is by far the most challenging stage. As I said, a prospective exploration study is often a bridge to a randomized controlled trial. And one of the things is to work out what is the question you want to ask in a randomized controlled trial. Do you have a group of surgeons that have an idea that they can put together to make a trial? It's that PICO questions you have to ask for a trial. Can you work out what that's going to be? Again, you'll get some data you can use for power calculations for a larger study. You also can learn about learning curves and learning curve data, and see how many, do you get an idea of how many cases or what has to be done before people can be said to be competent in doing the procedure. And during the procedure, you want to first of all define what the procedure really is, and also sort out the quality assurance and outcome measures. And again, I mentioned before, the qualitative element is really helpful in to work out what maximizes recruitment. if you're trying to recruit someone to a new study, one of the techniques, for example, is to record your consent of patients when you're consenting patients. And you can look at what you're saying to patients and pick out words or things that patients may respond to that you might want to avoid for future consenting of patients. This has been a very useful thing for other studies. By the time you reach stage 3, the assessment stage, this is more like what we know from the phase 3 drug studies. This is where you have sufficient surgeons with experience, and again, you can do a multi-center study comparing this new procedure, comparing with the current standards of care. And this is commonly done through a randomized controlled trial, just as you would find in a drug trial. So this is the time when you could think about looking at doing an assessment through a randomized controlled trial. The next stage, stage 4, is the long-term follow-up stage. And this is basically a quality assurance that aims to look at the long-term outcomes and also gets an idea about changes or drift in indications over time. So you often find that you start doing something with a very clear indication, and over time, because you get used to it, you say, oh, let's just try it on different groups of patients. And you get an idea of whether things have drifted from the original group that you had originally done, the trials originally showed was successful. And this can be done through, certainly registries are really important for this, to maintain this data and collect this information, but also you can set up and you can run in randomized trials nested within a comprehensive registry, and also run some observational studies. I mentioned earlier about radiotherapy. Well, there's now been developed an ideal R for radiotherapy. And this uses the same stages. Stage 0 is a preclinical stage, which is how will the technology work for patients. It can do with uses in phantoms. It can be do with planning studies or delineation studies, depending on what you're doing with this. Stage 1 is then a first-in-man technical report. Stage 2A is the technical optimization. How can you make the technique better? And again, it's the same principles of this reporting all the cases you use prospectively, and talk about changes in the technique that you use as you go through this. Stage 2B is the proof of early clinical effectiveness and safety. And again, this is where you start moving it out into a more of a multi-center setting before going on to the stage 3, which is the formal comparison with the standard treatment, usually as a randomized controlled trial, before looking at the long-term outcomes and long-term surveillance. The other thing the ideal collaboration is trying to do is promote this as good practice for surgical trials. So the idea is they're trying to get journal editors to accept ideal studies, both as protocols and also as full reports, to get the regulators to link approval, particularly for devices, to ideal stage studies, to get professional societies to support this model within their guidelines, and also to possibly develop registries if it's applicable, and also to get the research funders to support these ideal studies and understand what they are. Because I've been to grant research funders, I've been to grant giving bodies where I've pitched for an ideal study, and at the moment people don't really know enough about them to be able to provide a lot of information. So in summary, the role of surgeons in clinical trials, well, to do with tissue sampling. Where do you want the tissue from? It's really important that you get the tissue and it's properly annotated where the samples come from. Image guidance and fluorescence can help you do that. There is complexity amongst surgical trials, as well as radiotherapy or rehabilitation studies that are not like drug studies. There are very few drug studies where there is an issue about how you give the drug in the first place. The ideal framework and the ideal collaboration gives an idea of how we should be looking at these surgical interventions. And just as a side issue is that organizations, and I think eEANO is one of these organizations, we should be encouraging ideal type studies, using these ideal principles, and we should be discouraging the use of case series.